Hello friends, welcome to Midlife. Today we will continue with the same topic of inflammation, that is cellular events. So, the main function of the increased vascular permeability and stasis is to deliver leukocytes, that is WBC, at the site of injury. Here you can see this is the blood vessel and there is a microbe who has entered just below the skin. Microbe is extravascular and you can see inside the blood vessel WBC is present, and we have already created gaps here by one of the following 5 mechanisms. Now question arises, what is the problem? We can directly take the WBC out and make a fight with the microbe. So it doesn't happen this way. Have you heard about Bernoulli's theorem? So it states that in a hollow pipe, so this is a pipe, whenever any fluid is traveling, the heaviest portion of the fluid will travel along the center and the lightest portion of the fluid will travel along the periphery. So as you know, in our blood there are 4 things, that is RBC, WBC, platelet and plasma. Heaviest among them is WBC. So the heaviest among them is WBC, so it travels along the center. Surrounding WBC is present RBC. Surrounding RBC there is presence of platelets, and surrounding platelets there is presence of plasma, which is the lightest. So this is known as axial blood flow. Axial blood flow. Which is normal. Our blood flows like this. Now there is hole in the periphery. So what has to come out is at center. So how it will directly come out of the gap? Now let's see this figure. Here all the 6 steps of cellular events has taken place. As we know that the first step is known as margination and pavementing. Second step is known as rolling. Then after adhesion, transmigration, chemotaxis, and then lastly phagocytosis. So now let's see. The first thing, that WBC has to leave the center and it has to travel towards the margin or periphery of the blood vessel, and this process of leaving the center and moving towards the periphery of the blood vessel is known as margination, and this is also known as reverse axial blood flow. That is, everything is coming towards the periphery due to acute inflammation. Now since holes are present at distance from WBC, so now WBC will start rolling at the periphery, forming transient bond with the endothelial cells. And second step is known as rolling. Now after it approaches the gaps, after it approaches towards the gap, it has to stop. So the third step is known as adhesion. After adhesion, WBC will come out of the gap or blood vessel, leaving intracellular space and coming into the extravascular space here. and this process is known as transmigration. Now after coming out, it has to fight with the microbe, and for this microbe will release some chemicals or secrete some chemicals which will attract this WBC towards the microbe, and this process is known as chemotaxis. Now after reaching towards the microbe, the WBC will eat the microbe, and this process is known as phagocytosis. So what are the 6 steps of cellular events? Tell me in the comment section. As we have already discussed the 6 steps of cellular events in the diagram, now let's see its note one by one. So the first step of cellular event was margination. So the normal axial flow consists of central stream of cells comprised by leukocytes and RBCs and peripheral cell free layer of plasma close to vessel wall. WBCs leave the center and comes at periphery of blood vessel, which is also known as reversal of axial blood flow. Now let's see this flow chart. So due to increased vascular permeability, there was the formation of gaps, as we have already studied this. So due to this, there was slowing or stasis of the blood, that is central stream of cells widens, and due to the gap there was the leakage of the fluid as well as protein, that is this step was known as exudation. So loss of plasma by exudation, peripheral plasma zone becomes narrower. As a result of this redistribution, neutrophils of the central column comes close to the vessel wall, also known as reversal of axial blood flow, and this process is known as margination and pavementing. The endothelium can be virtually lined by white cells called as pavementing. Now let's see the second step known as rolling. So peripherally marginated and pavemented neutrophils slowly roll over the endothelial cells lining the blood vessel wall due to transient bonds between them. This is known as rolling. Now the third step is known as adhesion. This is followed by transient bond between the leukocytes and endothelial cells becoming firmer. So this was known as adhesion. Now let's study about the mechanism of rolling and adhesion. So basically there are 5 pairs of molecules due to which the endothelial cells and WBC forms bond within each other. So these molecules are known as CAM, that is complementary adhesion molecules. So the attachment of leukocytes to endothelial cells is mediated by complementary adhesion molecule, that is CAM, on the 2 cell types. So the following CAM molecules bring about rolling and adhesion phases. So here is the table of molecules and their role. So the molecules of endothelial cells are P-selectin, E-selectin, glycam-1, also known as CD34, ICAM-1, known as immunoglobulin family, BCAM-1, it is also of immunoglobulin family, and lastly PCAM-1, that is CD31. And also one molecule of selectin is L-selectin, which is not molecule of endothelial molecule. It is the molecule present in WBC, that is leukocyte molecule. And PCAM1, that is CD31, is present on both WBC as well as endothelial cell. So here the P-selectin of endothelial molecule make bond with sialyl Lewis X modified protein of leukocyte molecule. E-selectin make bond with sialyl Lewis-X modified protein. So it is easy to remember. Both P and E selectin of endothelial molecules make bond with sialyl Lewis-X modified protein of leukocyte molecule. Now here comes L-selectin, which is the molecule of WBC, not endothelial cell, and it will make bond with glycam-1, that is CD34 molecule of endothelial cell. Now comes ICAM and BCAM. So remember in this way, ICAM1 will make bond with beta-2 integrin and BCAM1 will make bond with beta-1 integrin. So beta-2 integrin is also known as CD11 or CD18, LFA1 or MAC1, and beta-1 integrin is also known as VLA4. Now let's see its role or function. So the function of P-selectin is rolling, E-selectin is rolling and adhesion. Function of glycam-1 is also rolling, and the function of ICAM and BCAM-1 is adhesion, and the function of PCAM is diapetesis, that is transmigration. And in the bondage of P-selectin and sialyl Lewis-X modified protein, neutrophils, monocytes and T lymphocytes play a major role, whereas in E-selectin, neutrophils and monocytes are present as well as T lymphocytes, and in glycam-1, neutrophils, monocytes are present, whereas in ICAM there is neutrophils, monocytes and lymphocytes, whereas in BCAM-1 there is presence of eosinophils, monocyte, lymphocytes are present, whereas can you see this diagram? On the endothelial lining, 5 pairs of molecules are present. Here we must know which molecule is present on endothelium and which molecule is present on WBC. So there are P-selectin and E-selectin, which is present on endothelium, and it forms bond with sialyl Lewis-X protein molecule, which is present on WBC. And L-selectin molecule, which is present on WBC, will form bond with glycam, and ICAM and VCAM will form bond with beta-2 and beta-1 integrins respectively, and PCAM molecule will help in transmigration of WBC, that is diapetesis. It is present on both sides, that is on endothelial cell as well as WBC here. Now let's see how transmigration takes place. So receptors are present on endothelium, these are the receptors, and also on corresponding WBC. Here, these are the receptors on both sides, which pushes the WBC towards downward out of the gap. So what are the receptors? These are PCAM, which helps in transmigration or diapetesis. So transmigration is the movement of WBC from intravascular to extravascular space with the help of PCAM receptor, also known as CD31. Now let's see this. Now remember that P-selectin, also known as CD64P. It is present on endothelial cells and platelets, and it is involved in rolling. E-selectin, also known as CD64E, it is also present on endothelial cells and it is associated with both rolling as well as adhesion. L-selectin, also known as CD62L, is present on lymphocytes and neutrophils, which is responsible for rolling. Now P and L-selectin are involved in rolling, whereas E-selectin is involved in both rolling as well as adhesion. Now the intercellular adhesion molecule, that is ICAM1, also known as CD54, it is present on endothelial cell, and vascular cell adhesion molecule 1, that is VCAM1, also named as CD106, is also present on endothelial cell. Platelet endothelial cell adhesion molecule 1, that is PCAM1 or CD31, is present on both endothelial cell and leukocyte, which is involved in leukocyte migration from endothelial surface, that is known as transmigration or diapetesis. Now, after completing the mechanism of rolling and adhesion, now let's come back to the fourth step of cellular event, that is transmigration, also known as diapetesis. Here it is mistake in the spelling of diabetes. So ignoring that, let's continue. Escaping out of WBC into the extravascular space is known as transmigration. After sticking of neutrophils to endothelium, they move along the endothelial surface till a suitable site between the endothelial cells is found where the neutrophils throw out cytoplasmic pseudopores. Then the neutrophils lodges between the endothelial cells and basement membrane. After that, it crosses the basement membrane by damaging it with secreted collagenases. After that, it escapes out into the extravascular space, and this is known as transmigration. Now, simultaneous to immigration of leukocytes, escape of red cells through gaps between the endothelial cells is known as diapetesis, which is also a passive phenomenon, and PCAM is a receptor which helps in the process of transmigration. Remember, neutrophils are the dominant cell in the first 24 hours. After that, monocyte or macrophages appear in the next 24 to 48 hours. However, neutrophils are short-lived from 24 to 48 hours, which monocyte and macrophages survive much longer. Now the fifth step of cellular event is known as chemotaxis. After the WBC comes in extravascular space, the microbes secrete some chemicals which attract the WBC towards the microbe, and after that there will be phagocytosis. So this process of secreting chemical and the attraction of WBC towards the microbe is known as chemotaxis. So what are those chemicals? So the four chemicals are secreted by the microbe, and those are known as LTB4, C3A, C5A, which is most potent, and IL-8. So chemotaxis is a unidirectional oriented along a chemical gradient. Now potent chemotactic substances are leukotriene B4, that is LTB4, components of complement system, that is C5A and C3A, cytokines, that is interleukins in particular, that is IL-8, and fourth one is soluble bacterial products such as formulated peptide. C5A is the most powerful chemoattractant chemokine. Locomotion in chemotaxis requires polymerization. that is assembly of actin. Now let's see this Boyden's chamber experiment. A millipore filter of 3 μm pore size separates the suspension of leukocytes from the test solution containing chemotactic agent. Leukocytes migrate through the pores of filter towards the chemotactic agent. And this shows movement of neutrophils towards chemotactic agent, that is C5a. Now let's do some Q&A. So let's proceed with question 1. The correct sequence in extravasation of leukocyte is A. Margination, rolling, adhesion, transmigration. B. Transmigration, margination, rolling, adhesion. C. Rolling, adhesion, transmigration, margination. D. Adhesion, transmigration, margination and rolling. So what is the correct answer? So the correct answer here is A, that is margination, rolling, adhesion and transmigration. Let's proceed towards the question 2. Leukocyte migration through capillary wall is known as A. Rolling, B. Diapedesis, C. Migration, D. Pavementing. So what is the correct answer? The correct answer is known as diapedesis. Question 3. Rolling of leukocytes on endothelial cells is mediated by A. ICAM1, B. beta-2 integrin, C. IL-8, D. P-selectin. So here the correct answer is D, that is P-selectin. Question 4. Most important for diabetes is A. PCAM, B. Selectin, C. Integrin, D. Mucin-like glycoprotein. So the correct answer is, here the correct answer is A, that is PCAM. Now let's see question 5. All of which is family of selectin except A. P-selectin, B. L-selectin, C. A-selectin, D. E-selectin. So what's the correct answer? It is C, that is A-selectin. We have already discussed about P-selectin, E-selectin and L-selectin. Now let's see question 6. The most important mediator of chemotaxis among following is A. C3b, B. C5a, C. C567, D. C2. So what's the correct answer? The correct answer here is B, that is C5a. Now let's see question 7. Chemotaxis in response to activation of cells results in A. Random multidirectional movement, B. Unidirectional motion, C. Adhesion to endothelium, D. Augmented oxygen dependent bactericidal effect. So what's the correct answer? The correct answer here is B, that is unidirectional motion. Now let's see question 8. Chemotaxis is mediated by A. Histamine, B. Leukotriene B4 and C5a, C. Leukotriene C4 and C3a, D. Bradykinin. So the correct answer is, here the correct answer is B, that is Leukotriene B4 and C5a. Now let's see the last step of cellular event, that is known as phagocytosis. So the process of engulfment of microbes by the WBCs, that is cell eating, is known as phagocytosis. Cells performing this function are known as phagocytes, and there are two types of phagocytic cell. One is polymorphonuclear neutrophils, that is PMNs, which appear early in acute inflammatory response, and secondly is monocytes, commonly known as macrophages. The sequence of phagocytosis was appreciated by Mechnikov. 1. Recognition and attachment. 2. Engulfment. 3 is killing and degradation. So this was all about the 6th process of cellular event. If you have any queries, please let me know in the comment section. Please like, share and subscribe for more videos of pathology. Thank you.